Today we're going to discuss how the new Wegovy pill works by looking at its active ingredient, oral semaglutide. Semaglutide was originally developed to help manage type 2 diabetes by targeting blood sugar control. There is an oral form of semaglutide used for diabetes called Rybelsus. Rybelsus is typically taken at doses under 10 milligrams per day, which is sufficient to improve glucose control in type 2 diabetes. Higher drug exposure is needed to reduce appetite and support sustained weight loss. Semaglutide has been available for weight management for several years, but until now only in injectable form. The FDA has now approved an oral formulation for weight reduction under the brand name Wegovy, which delivers up to 25 milligrams daily. In short, the same molecule, semaglutide, has been fine-tuned for different indications with dosage and formulation tailored to its therapeutic goal. So what exactly is the Wegovy pill? Wegovy is the brand name of the medication. It contains semaglutide, the active drug inside the pill. Semaglutide works by mimicking a naturally occurring hormone called GLP-1, short for glucagon-like peptide 1. GLP-1 is a gut hormone released after eating. It helps regulate appetite, slows gastric emptying, and enhances insulin secretion. At the molecular level, GLP-1 is a peptide made up of a specific sequence of amino acids. Peptides like GLP-1 are normally broken down quickly and are generally not well absorbed when taken by mouth. Semaglutide is also a peptide, and it is closely related in structure to natural GLP-1. The structural modifications highlighted here stabilize the peptide so it lasts much longer in the body, while still activating the same GLP-1 receptor. Wegovy, therefore, contains a carefully modified peptide that behaves like GLP-1 but is engineered to work as a medicine. So why do peptides not make good oral drugs? First, peptides are treated like food and are digested in the stomach. When a pill arrives in the stomach, it dissolves, releasing the peptide into the stomach contents, where digestive enzymes rapidly cleave peptide bonds and break them down. Second is poor intestinal absorption. Even if intact peptides make it through the stomach to the small intestine, they struggle to cross the intestinal lining. They are large, polar molecules with no easy way to escape the gut lumen, cross the interstitial space, and enter the circulation. And even if a small amount is absorbed, the liver and kidneys clear peptides from the circulation very quickly. That is why most peptide drugs are given by injection. To overcome these limitations, peptides can be co-formulated with an absorption enhancer. An absorption enhancer is a functional excipient that increases peptide penetration across epithelial barriers. The Wegovy pill combines semaglutide with SNAC, which stands for sodium N8 2-hydroxybenzoyl amino caprolate. This molecule has several key properties. First, it helps protect semaglutide from the harsh gastric environment. SNAC contains a carboxylate group and a phenolic hydroxyl group. In the acidic conditions of the stomach, these groups participate in acid-base equilibria that buffer the local pH. As the Wegovy tablet dissolves, SNAC is released in large molar excess, immediately next to semaglutide, creating a local microenvironment. This buffered microenvironment reduces acid-mediated peptide degradation and suppresses gastric protease activity, helping to stabilize semaglutide long enough for absorption to occur. Second, SNAC facilitates absorption. SNAC is amphiphilic, meaning it contains both hydrophilic and hydrophobic regions. This allows it to interact transiently with the lipid bilayer of gastric epithelial cell membranes, temporarily disrupting the membrane structure and increasing membrane fluidity. As a result, large polar molecules such as peptides can cross the epithelial cell more readily via a transcellular route. Semaglutide can then move across the epithelium and enter the interstitial space, from where it gains access to the circulation through nearby capillaries. In this way, SNAC enables semaglutide to be absorbed directly from the stomach, bypassing the intestine where protease activity is higher and peptide permeability is even more limited. Importantly, SNAC acts only locally. It does not alter the pH of gastric contents as a whole, and its effects on membrane permeability are transient. As SNAC is diluted, normal gastric conditions are rapidly restored. Oral and injectable Wegovy contain the same active drug, semaglutide, and they work by activating the same GLP-1 receptor. The key difference lies in how the drug enters the body. Injectable semaglutide is administered subcutaneously, meaning it is injected into the tissue under the skin. From there, the drug is slowly absorbed into the bloodstream. Because this route bypasses the gastrointestinal tract, semaglutide avoids digestive enzymes and the barriers to absorption seen with oral dosing. As a result, a large proportion of the injected dose eventually reaches the circulation, giving it high bioavailability and allowing it to be administered at a relatively low dose. In contrast, oral semaglutide taken as a daily dose must survive the stomach and cross the gut lining before it can be absorbed. Even with an absorption enhancer, only a very small fraction of the swallowed dose reaches the circulation. This low bioavailability means that much higher daily doses are required to achieve the same overall drug exposure. Oral semaglutide offers the advantage of avoiding injections, which many patients prefer. However, it must be taken under strict dosing conditions to work effectively. The tablet is taken on an empty stomach with a small volume of water, and no food, drink, or other medications should be consumed for at least 30 minutes afterward. These conditions are necessary to prevent dilution of the absorption enhancer and to preserve the local environment that allows semaglutide to be absorbed. Injectable semaglutide provides more predictable drug levels and requires less frequent dosing, but involves injections which some patients find inconvenient or uncomfortable. In short, the oral dose is much higher, not because the drug is weaker, but because the delivery route is less efficient. The choice between oral and injectable semaglutide reflects a balance between convenience, reliability of absorption, and patient preference. Once semaglutide has been absorbed into the bloodstream, either from a tablet or from subcutaneous injection, it circulates throughout the body. At this point, the route of administration no longer matters. What matters is where the drug goes and which receptors it activates. Semaglutide is a GLP-1 receptor agonist, meaning it mimics the actions of the natural hormone GLP-1. GLP-1 receptors are distributed across multiple organs, so semaglutide does not act in just one place. In the brain, particularly in appetite-regulating centers of the hypothalamus, GLP-1 receptor activation increases satiety and reduces hunger. This central effect is a major contributor to reduced food intake and weight loss. In the gastrointestinal tract, GLP-1 signaling slows gastric emptying. Food leaves the stomach more slowly, which enhances feelings of fullness after eating and blunts rapid rises in blood glucose. In the pancreas, GLP-1 receptor activation enhances insulin secretion, but only when blood glucose levels are elevated. And at the same time, it suppresses glucagon release. This glucose-dependent action helps improve glycemic control while limiting the risk of hypoglycemia. Together, these effects explain why semaglutide influences both blood glucose regulation and body weight. It acts as a coordinated hormonal signal linking the gut, the brain, and the pancreas. In the next slides, we'll zoom in on one of these targets and look more closely at how GLP-1 affects pancreatic function. The pancreas is a lobular gland situated just behind the stomach. The lobules contain the structures that synthesize the substances that are key to the function of the pancreas. Around the outer circumference of each lobule are the acini, which synthesize digestive enzymes. The acinar cells making up each acini secrete the enzymes into a central duct. It connects to the main pancreatic duct, which delivers the enzymes to the inside of the small intestine, where they work to break down carbohydrates, proteins, and fats in food. This is an example of an exocrine gland, which secretes directly into a ductal system. The central core of the lobule is known as the endocrine pancreas. It is comprised of clusters of secretory cells known as an islet of Langerhans. There are several cell types, each specialized for secreting a particular hormone. The majority secrete insulin and are known as beta cells. They account for 40 to 50% of islet cells in humans. Glucagon secreting cells are called alpha cells and make up about 20% of the islet cell population. The other hormones secreted are somatostatin from delta cells, pancreatic polypeptide from gamma cells, and ghrelin from epsilon cells. They, respectively, make up less than 10%, 5%, and 1% of the total cell population. The cells in the islet secrete their hormones out of the cell and into the interstitial space. There is an extensive capillary network supplying the islets with blood, so the secreted hormones are taken up into the circulation, which transports them to their site of action. The hormones insulin and glucagon play key and complementary roles in the regulation of blood glucose. They form negative feedback loops to maintain the blood glucose concentration in its optimal range of 4 to 5.9 millimolar. When blood glucose levels rise following a meal, the increase is detected in the beta islet cells of the pancreas as blood flows through the organ. The beta cells respond by releasing insulin into the circulation. Insulin acts on several sites in the body to remove glucose from the blood. It stimulates adipocytes in adipose tissue to take up and store excess glucose as fat. In the liver and skeletal muscle, insulin stimulates cells to take up and store excess glucose as glycogen. These effects lower the blood glucose and restore it back to the normal level. When blood glucose is too low, alpha cells in the pancreas release the hormone glucagon. It acts on the liver to stimulate the breakdown of glycogen into glucose, which enters the circulation and restores the glucose level to normal. So how does GLP-1 exert its effects on insulin and glucagon secretion? Both endogenous GLP-1 and semaglutide act as agonists at the GLP-1 receptor. This means they bind to specific receptor sites on the cell membrane and activate those receptors to trigger a cellular response. The GLP-1 receptor is a G-protein coupled receptor, or GPCR. These are membrane-spanning proteins that interact on the cytoplasmic side with a heterotrimeric G protein composed of 3 subunits: alpha, beta, and gamma. In the inactive state, the alpha subunit is bound to GDP. When an agonist binds to the receptor on the extracellular surface, it induces a conformational change in the receptor. This promotes the exchange of GDP for GTP on the alpha subunit of the G protein. Once GTP is bound, the alpha subunit dissociates from the beta and gamma subunits and becomes free to interact with downstream signaling proteins. There are several types of G protein alpha subunits. The GLP-1 receptor couples predominantly to the GS isoform, which stimulates the enzyme adenyl cyclase. Adenyl cyclase increases the conversion of ATP into cyclic AMP. Rising intracellular cyclic AMP then activates protein kinase A, as well as other cyclic AMP-sensitive signaling pathways. In pancreatic beta cells, activation of protein kinase A leads to phosphorylation of multiple downstream targets. These include the transcription factor CREB, which promotes insulin gene transcription, as well as proteins involved in insulin granule trafficking and exocytosis. Together, these effects enhance glucose-dependent insulin secretion. Cyclic AMP signaling activates additional pathways, including phosphatidylinositol 3 kinase and the kinase AKT. These pathways increase beta cell responsiveness to glucose and support beta cell function and survival over time. The situation is more complex in pancreatic alpha cells, where the overall physiological effect of GLP-1 is suppression of glucagon secretion. GLP-1 receptor activation in alpha cells increases intracellular cyclic AMP and engages downstream effectors such as protein kinase A and EPAC, the exchange protein directly activated by cyclic AMP. In isolation, these signaling pathways would be expected to stimulate glucagon release rather than inhibit it. However, GLP-1 receptor expression in alpha cells is relatively low, and direct signaling in alpha cells appears to be only part of the overall response. An important additional mechanism involves delta cells within the islet. GLP-1 receptors are expressed on delta cells where receptor activation stimulates the release of somatostatin. Somatostatin then acts in a paracrine manner on neighbouring alpha cells to inhibit glucagon secretion. As a result of this indirect intra-islet signalling, the net effect of GLP-1 receptor activation is a reduction in glucagon release, particularly when blood glucose levels are elevated. These same receptor pathways explain both the therapeutic effects and side effects of semaglutide. In the brain, GLP-1 receptor activation in appetite-regulating centers reduces hunger and increases satiety. This central effect is a major driver of reduced food intake and sustained weight loss. In the gastrointestinal tract, GLP-1 signaling slows gastric emptying. Food remains in the stomach for longer, which enhances feelings of fullness and helps limit meal size. This delayed gastric emptying also contributes to improved post-meal glucose control. In the pancreas, GLP-1 receptor activation enhances glucose-dependent insulin secretion and suppresses glucagon release, supporting glycemic control without increasing the risk of hypoglycemia. However, these same physiological effects also explain the most common side effects of GLP-1 receptor agonists. Slowed gastric emptying and enhanced satiety signaling can manifest as nausea, abdominal discomfort, or vomiting, particularly when drug exposure rises quickly. GLP-1 receptors in the brainstem are also involved in nausea signaling, further contributing to these symptoms. Importantly, these effects are not off-target or toxic. They represent an exaggeration of the normal actions of GLP-1. This is why side effects are dose-dependent and why gradual dose escalation improves tolerability. In essence, the benefits and side effects of semaglutide arise from the same biology. The goal of treatment is to achieve sufficient GLP-1 receptor activation to support weight loss while minimizing excessive stimulation that leads to intolerance. When oral Wegovy treatment begins, the dose is started low and increased gradually over time. This stepwise approach, known as dose titration, is essential for tolerability. If exposure to semaglutide rises too quickly, the body has little opportunity to adapt, and side effects are more likely to occur. Gradual dose escalation allows the gastrointestinal tract and the central nervous system to adjust to sustained GLP-1 receptor stimulation. Over time, tolerance develops to the nausea-inducing effects, while the beneficial effects on appetite regulation and glucose control are largely preserved. For oral Wegovy, titration is especially important because higher daily doses are required to overcome low bioavailability. Increasing the dose slowly helps patients reach an effective dose while minimizing side effects and improving long-term adherence. In short, gradual dose increases strike a balance between achieving sufficient GLP-1 receptor activation for clinical benefit and minimizing side effects that could otherwise limit adherence to treatment. To summarize, the Wegovy pill contains semaglutide, a GLP-1 receptor agonist that mimics a naturally occurring gut hormone. The biology underlying its effects is well established and is the same whether semaglutide is given by injection or by mouth. What is new is the formulation. By combining semaglutide with the absorption enhancer SNAC, it has become possible to deliver a peptide drug orally, despite the challenges of degradation and poor absorption in the gastrointestinal tract. The low efficiency of this process explains why higher daily doses are required. Once absorbed, semaglutide acts systemically on GLP-1 receptors in the brain, gastrointestinal tract, and pancreas. These coordinated effects reduce appetite, slow gastric emptying, and improve glucose regulation. The same mechanisms that drive weight loss also explain the characteristic side effects, which is why careful dose titration is essential. In short, oral Wegovy works not because the drug itself is new, but because advances in formulation science have allowed a well-understood biological pathway to be harnessed in a new way.